Assalamu alaikum wa rahmatullahi wa barakatuh. Hello everyone, welcome to lecture 15, Organic Chemistry 3, Carbohydrates and Nucleic Acids. Nucleic acids, DNA deoxyribonucleic acid and RNA ribonucleic acid are polymers of nucleotides linked in a chain through phosphodiester bonds. In biological systems, they serve as information-carrying molecules, or in the case of some RNA molecules, as a catalyst. In a typical cell, DNA is found primarily in the nucleus, where it carries the permanent genetic code. The molecules of DNA are huge, with molecular weight up to 50 billion. When the cell divides, DNA replicates to form two copies of the daughter cells. DNA is relatively stable, providing a medium for transmission of genetic information from one generation to the next. RNA molecules are typically much smaller than DNA, and they are more easily hydrolyzed and broken down. Before discussing the structure of DNA and RNA, we need to know what are the components of its structure: bases, nucleosides, and nucleosides. Bases: a nitrogen-containing base, either pyrimidine 1 ring or purine 2 rings. Here is a pyrimidine bases: uracil, cytosine, thymine, and the purine base, the fused 2 rings, adenine and guanine. Well, each has at least one NH site at which an organic substituent may be attached. Okay? They are polyfunctional bases and may exist in tautomeric forms. Cytosine and uracil have tautomeric forms with aromatic hydroxy groups. Draw these tautomeric forms. Well, looking for cytosine, this is a canonical forms which chemists use often. Now we can move the 2 electrons in here, getting hydroxy group as substituent. We call this amino hydroxyl form. In here we have here amino oxo. Here we have imino oxo. Here the hydrogen bond NH goes down, and this nitrogen will carry the hydrogen. And we can do another form, imino hydroxyl, where again the hydrogen relocate on the oxygen. For uracil, well, we can have the OH and double bond. In here we can have again this one moved in here, 2 electrons, transferring the hydrogen from the nitrogen to oxygen. Or we can have this oxo and this hydroxy. You can draw more than that, okay, but these are the well-known stable forms. Okay, the combination of a base and sugar, ribose or deoxyribose, is called nucleoside. Here is beta deribofuranose structure, replacing the OH with nitrogen containing heterocycles, we call it ribonucleoside. As an example, adenosine, where we replace the OH with adenine as a base. An aliphatic aminoglycoside is relatively stable to base, but it is quickly hydrolyzed by dilute acid. Propose a mechanism for the acid catalyzed hydrolysis. So we have here aminoglycoside in acidic media. They split this molecule to ribose and amines. Of course, in acidic media will be ammonium salt. Ribonucleosides are not so easily hydrolyzed, requiring relatively strong acid. Using your mechanism for this part, the above part, show why cytidine and adenosine, for example, are not so readily hydrolyzed. Explain why this stability is important for living organisms. Well, the first step that the aminoglycoside will gets protonated on the N nitrogen atom to form ammonium salt. The lone pair of oxygen will assist the amino group to leave as a good leaving group, forming carbocation intermediate, which is stabilized by resonance. Then attacked by the water oxygen molecule to form the sugar. Of course, the OH could attack, water molecule will attack from the above or from the bottom face to form the alpha and beta sugar. Nucleosides are less rapidly hydrolyzed in aqueous acid because the site of protonation is much less basic than aliphatic amine in the aminoglycoside. Nucleoside requires stronger acid, longer time, higher temperature to be hydrolyzed. Here in the cytidine, we have the lone pair. This lone pair will be delocalized over the oxygen, forming positive charge, negative charge on oxygen. This lone pair is actually the site for protonation, so it gets protonated, and again this lone pair will push the heterocyclic out, cleaving the CN bond. In a living system, the hydrolyze would cause damage or even death of an organism if its DNA or RNA were easily decomposed. In adenosine, we have nitrogen 1, 3, and 7 and 9. Again, we will find out that the better candidate for attachment are N1 and 3 and 7, not 9. The experiment showed that the site preferred is N1 over N3 and N7. Okay, you can find out that in the Journal of Biochemistry, published in 1970, page 4907. Okay, what are nucleotides? Nucleotides are the building blocks of all nucleic acids. Nucleotides have distinctive structure composed of three components, covalently bonded together: nitrogen-containing base, either pyrimidine or purine, and 5-carbon sugar ribose or deoxyribose, as well as phosphate groups. So we have here sugar, phosphates, and base. That's ribonucleotide. Ribonucleotide. Here we have deoxyribose. We don't have here OH on this carbon. Again, we have deoxyribose. We have the phosphate, and we have the base. Nucleotides also exist in activated forms containing 1, 2, or 3 phosphates, called nucleotide, monophosphate, diphosphate, or triphosphates. If the sugar in the nucleotide is deoxyribose, the nucleotide is called deoxynucleotide. If the sugar is ribose, the term ribonucleotide is used. The acidic character of the nucleic acid was attributed to the phosphoric acid moiety. So we have the sugar and the phosphate. In acidic media, we have these OHs protonated. In nearly neutral medium, we have monoanion. In basic media, we have the dianion. There are 5 common bases, and 4 are generally represented in either DNA or RNA. Those bases are adenine, guanine, cytosine, thymine, uracil. Adenine present in adenosine RNA or deoxyadenosine in DNA. Symbol capital A. Guanine as a base present in guanosine and deoxyguanosine. Symbol capital G. Cytosine, capital C as a symbol, present in cytidine in RNA and deoxycytidine in DNA. Thymine, capital T, present only in deoxythymidine in DNA, while uracil present in uridine in RNA. So, as you can see, adenosine, guanine, cytosine, thymine present in DNA, while adenine, guanine, cytosine, uracil presents in RNA. A key feature of all nucleic acids is that they have 2 distinctive ends, the 5' and 3' ends. These 2 ends refer to the 5' and 3' carbon on the sugar part. For both DNA and RNA, the 5' end bears a phosphate and the 3' end hydroxy group. Another important concept in nucleic acid structure is that DNA and RNA polymerases add nucleotides to the 3' end of the previously incorporated base. Another way to put this is that nucleic acids are synthesized in 5' to 3' direction. Well, here in RNA synthesis, we have 2 nucleotides, one with terminal phosphate on 5 prime and the other one free OH on 3 prime. Elimination of water, link them together through the phosphate to form dinucleotide. Here we have the 5 prime end, and here we have the 3 prime end. Most DNA exists in the famous form of double helix, in which two linear standards of DNA are wound around one another. The major force promoting formation of this helix is complementary base pairing. Adenine form hydrogen bonds with thymine. Hydrogen bonds in here, 2 hydrogen bonds. Adenine, thymine. And guanine form hydrogen bonds with cytosine. But here we have 3 hydrogen bonds. So here is more stronger. If we mix the 2 fragments, ATGC and TACG, together, the following duplex will form. The A and T and A are linked together through hydrogen bonds. G and C, C and G, again, here we have 3 hydrogen bonds, 3 hydrogen bonds. Here we have 2 hydrogen bonds. So the 2 standards are arranged anti-parallel, 5 prime to 3 prime. Here we have the 3 prime and 5 prime. So they are matching with one another through the hydrogen bonds. Again, the CG bond with 3 hydrogen bonds will be stronger and needs higher energy to unzip. Watson and Crick found that the 2 complementary strands of DNA are coiled into a helix conformation about 20 angstrom in diameter, with both chains coiled around the same X. The helix makes a complete return for every 10 residues, or about 1 turn in every 34 angstrom of length. For their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material, Crick, Watson, and Wilkins obtained 1962 Nobel Prize in Medicine. Erwin Chargaff's discovery that DNA contains equimolar amounts of guanine and cytosine, and also equimolar amount of adenine and thymine, has come to be known as Chargaff's rule, G equal to C and A equal to T. Does Chargaff's rule imply that equal amounts of guanine and adenine are present in DNA? That is, does G equal to A? Of course, the answer no. There is no relation between the amount of G and A. Does the Chargaff's rule imply that the sum of the purine residues equals to the sum of the pyrimidine residues? That is, does A plus G equal C plus T? The answer is yes, this must be true. Does Chargaff's rule apply only to double-stranded DNA, or would it also apply to each individual strand if the double helical strand were separated into its 2 complementary strands? The answer is Chargaff's rule must apply only to double-stranded DNA. For each G in one strand, there is a complementary C in the opposing strand, but there is no correlation between G and C in the same strand. Retroviruses like HIV, the pathogen responsible for AIDS, incorporated an RNA template that is copied into DNA during infection. The reverse transcriptase enzyme that copies RNA into DNA is relatively non-selective and error-prone, leading to high mutation rate. Its lack of selectivity is exploited by the anti-HIV drug AZT, 3' azido 2' 3' dideoxythymidine, which becomes phosphorylated and is incorporated by reverse transcriptase into DNA, where it acts as a chain terminator. Mammalian DNA polymerases are more selective, having a low affinity for AZT, so its toxicity is relatively low. Draw the structures of AZT and the natural deoxythymidine. Well, here is a AZT structure with azido on 3' and the natural deoxythymidine where the 3' has an OH. Draw the structure of AZT 5' triphosphate. So it will be the same structure. 3' has N3 azido group, and on 5' we have triphosphate. See you next lecture.